mentor here, Elisa Hovey, and my family in the back. And I work in Dr. Susan Mazur's lab, and we've been studying plant mating system evolution. And we've been trying to answer the question, why do some plants evolve mechanisms that make them able to self-fertilize? And to do that, we've been studying this flower called Clarkia Umbriculata. The common name for this flower is called Farewell to Spring. It's a flower that shows up just as summer begins, and it's all that's really left out in the Sierra Nevadas as far as flowering plants. And so the reason that we study Clarkia Umbriculata is because it is an outcrosser. An outcrosser is a plant that uses pollinators such as bees to transport pollen and sperm from the male sex organ to the female sex organ on another plant. And so outcrossing occurs between genetically distinct individuals, and it's dependent on pollinator vectors. But with Clarkia Umbriculata, these plants are also known to self-fertilize as well. And so it makes them a good model for understanding why do some plants develop these mechanisms. So self-fertilizing we refer to as selfing. A couple notes on these flowers here. If you look, some of the mechanisms that develop are to self-fertilize. Shown here in the closely related Exilus, which is a sulfur predominantly, is basically no spatial separation between the male and female sex organ, so that the pollen is readily available and it basically gets as much as it needs. And if you look at the outcrosser here, there's a wide degree of separation. There's also larger petals on the outcrosser because it has to attract its pollinator, the bee right here. And on the sulfur, there are smaller petals. The outcrossers flower a little bit later during the flowering season, and the sulfurs are known to flower earlier when conditions are more favorable and more resources such as water are available. So when you self-fertilize, there are possible risks. You risk producing unhealthy inbred individuals, and you also, as a population, you risk having low genetic variability. And so if some plague comes in, you don't have genetically distinct members of the population that will be able to have a resistance. That's one of the risks. But what is good about selfing is that you can reproduce independently of pollinators. And so we know from an article written by Jamie Fausto that habitat fragmentation and encroachment on habitats actually does affect plant-pollinator interactions, and that the more of that we continue to see in the future, the more adversely affected the relationship will be between plants and pollinators. Basically, you drive the pollinators away. So my objective was to look at something called pollen limitation. And why I'm looking at pollen limitation is because I want to know, does an insufficient deposition of pollen cause these plants to cope by developing selfing mechanisms? Do they evolve as selfers to compensate for the pollen that they're not receiving? So I was going to compare seed production between flowers or fruits of the same species, ones that receive additional pollen, such as the conditions experienced by a selfer, and ones that are just left out in natural conditions. And if we see a significant difference, then we can say, yes, there's an advantage to having a readily available amount of pollen to be able to produce the maximum number of seeds you can. And then I wanted to look at it early in the season and late in the season when conditions are drier and see if we are viewing the same effects, because we find the selfing plants that have evolved in the early season predominantly and the outcrossers later in the season. So we wanted to have some explanation for that. And of course, make some inferences based on the data that we find. So, little flower anatomy lesson. Each plant has multiple flowers. Those flowers, here's a flower right here, contain ovules. They're analogous to the human egg. And so pollen from the male part of the plant will transport sperm into that ovule, and a fertilized ovule will become a seed. And so when we want to see if pollen limitation is occurring, we look at seed production. Plants that are pollen limited will produce a lower ratio of seeds to ovules. So we went out in the field, Sierra Nevada. We went 3 times, 3 different locations, and we did treatments on 200 plants from each location. We treated 100 of them early and collected them, and then 100 late in the season when it's drier. On each individual plant that we went out and treated, we had a supplemented flower that was given pollen by hand. They have about 100 to 130 ovules, and basically we were caking on the pollen grains and the 500, so they were getting plenty. And then we have the other flower that's left in natural conditions. So that will tell us, if one produces a higher ratio of seeds to ovules than the other, then pollen limitation is occurring. Here's our 3 sites right here out in the southern Sierra Nevada. Awesome trip. I really enjoyed it. And then we allowed the flowers to mature into fruits, and we went out and collected these fruits by hand and brought them back into a lab, have them in test tubes. I dissected all of these, and in all, somewhere around 1,100 samples. So sample size, really great. We got some great statistics because we had a huge sample size. And that's the equation that we use to compare groups. Seed set is what we looked at. So we looked at the ratio of seeds to ovules that were originally in the plant. And then we recorded the data, and we used the program to analyze it using ANOVA, analysis of variance, and detect, was there a significant difference between the group that was hand-pollinated and the group that was not. And then we looked at early versus late season as well. And our results show that pollen limitation does occur. And so what we found is this y-axis, the seed set. The seed set is significantly greater in the supplemented group, both early in the season, as shown here. This is supplemented, this blue, supplemented is blue, and naturally pollinated is red. And in late, although early in the season, the seed set was significantly higher to fairly higher degree than late in the season. We saw more pollen limitation in the early season, even though we detected it in both seasons. Now, what's interesting about that, though, is we looked at something called abortion rates. Abortion is what happens when a plant has to allocate its resources to a fixed number of seeds. And so if you have dry conditions, the plant will, it'll say, okay, only these many seeds can grow. And so regardless of the pollen that you receive, you're getting a fixed number of seeds. And that's why I believe we didn't see a higher, we found pollen limitation more in the early season, because in the late season it's drier. And so the abortion rate would have masked the effect of pollen limitation anyway. So this is the natural group, supplemented group. You can see there's just a phenomenal difference between the rates here. So, conclusion, selfing is likely to have evolved under the influence of pollen limitation, and it does occur in the species we sampled out in Sierra Nevada, Clarkia Umbriculata. And we know that man-made disruptions of habitat can affect, can induce pollen limitation, and that it may drive the evolution of these plants in the future, and that high abortion rates are likely to have nullified the advantage of self-fertilization in the late season, because basically if you're not getting any more seeds when you self, then why evolve selfing? So I'd like to end by thanking my mentor, my parents, my lab partners, and all the INSET guys for this experience. I had a really great time. So, thank you.